for clarification. So are you taking like a whole brain approach in terms of like connectivity throughout the whole brain? Or are you probably doing like a network sort of approach and where you can look at connectivity like when you into
Right. That's
the
it.
networks of the limbic network?
I can jump in quickly if
I can jump in as well. I think because of the sample size, because it's 40, I think a whole network or whole brain analysis won't be very good to do in this case. I don't think we have enough power to do it. So it will probably be like a read like ROI to ROI approach.
Yeah, we haven't like looked
Yeah.
at the data set itself, and like there's not too much information on like open neural itself on like what regions they have indicated. So we'll probably have to take a look at that and see like whether or not they did collect like really specific like ROIs or XCs.
How did you get to this? Or is it like just? I think we're like very interested in this data set. And it's like, it's just very cool.
Speaking personally, I really like studies that use tasks. That's a part of my research that I do as part of my thesis, specifically emotional recognition tasks, social cognition tasks. So I think when we were kind of looking, originally actually wanted to look at a depressed population, but unfortunately there just wasn't a lot available. So ultimately we kind of decided to focus more on a task that we found interesting. Yeah. So kind of looking at it from a different perspective, I guess.
Also, it's like relatively new. Like we published it in like 2024 as well, and like it's a good sample size. Like 40 participants is pretty good, given we have like 2 weeks, like less than 2 weeks from now to get it done.
In the meta-analysis you mentioned earlier, which was the demographic the population they used?
I think I think yeah, it was so it was like mainly of studies in adults, I think, and youth. So it like looked at both.
Any other questions?
Yeah, for the task-based analysis. Are you doing like a task contract there, or are you just looking at connectivity within the task plan?
We're not sure yet about that, and we actually wanted to like get your opinion about what we can do like for those specific things. We were thinking of maybe like looking at connectivity and seeing how it differs between like positive and negative valence.
So like a connectivity, but within the task blocks.
Yeah.
Because there's like calm, depressed, like fearful, angry. So like within those.
Okay.
How long is the scan? Like, you know?
So it should be like for 30 seconds in total. So like each movie clip is 30 seconds. But then like each movie clip is separated by a 30-second white noise. So
Like 8 minutes.
But yeah, it's like around it, I think.
And it's the same for resting state? How long is the resting?
I don't know if they actually explicitly say it in the description. Yeah, because I looked, I didn't see that.
Probably be able to get that from the GSP.
And they only scan once for the task?
Yeah, it's a cross section.
That if you're taking a block of that scan, it might be very short. I mean, it would be a nice practice, but
it might like if you split the depending on how many blocks. Yeah, I guess you need to see how many TRs if you're trying to get TR specific to the task. You might not pulling to do your connectivity analysis. Yeah.
the whole task is like 8 minutes. The whole task scan.
How many events? Like how many types of events?
Like the different conditions.
There are 5 different
factors.
2 minutes. Yeah. Maybe short.
It might be short. It might still, might get something. I guess it depends on your TR too. It's 2 minutes, but
Yeah.
like if you have a short TR, then might come up in 2 minutes. Yeah, but you'd still learn how to know variance with the equation. they block or is that related?
I think it's cool.
Yeah.
Okay, blocks might help.
Because usually, so for resting now, like even 7, 8, which was standard a few years ago, now it's like considered too short
Yeah.
because the signal is kind of low. So for tasks, like if you're doing 3 minutes, just keep that in mind. That might not be.
Like when someone does something like sliding window, the windows. What's the little window?
I know.
It might be two, so we might kind of.
Yeah, like a few TRs.
Yeah.
Not much.
Okay.
Thank you.
Thank you.
You can get your cheesecake there. Everyone who presented get cheesecakes. Okay. Next group. Lorena, Grace, Hillary, Sam, Daniel.
It's only us three because Hillary is anyone.
Okay, yeah, I see. Hillary told me.
Do you want the chocolate or anything?
I would really want to have
Okay. Are you on Zoom?
like, yeah.
Sure.
Should have a look at it and especially like have my notes. That's anything wrong with the Wi-Fi on Thursday and it's loaded download and then you can't. Maybe like to look over this.
So it's Lorena, Lorena,
Is there a second page? going on?
Grace and Sam.
Well, I was in like, I think. This is the only page, right? Is there like final presentation around? Or no?
Yeah, we do.
Oh, okay.
So, speak loud. Speak loud.
Into.
Just in general. So we go in here. Here. On my computer. Yeah. We are recording.
I think this is.
I do.
Okay, we're ready for the next one. It's time for the next presentation.
Deep conversation.
Okay.
I've never tried cheesecake. Hey, everyone.
Hello. So here's our pitch for the BrainHack project. First, we'll start with introductions.
Hi, I'm Lorena. I just wrapped up my PhD in nutrition, so a little bit unrelated topic, but I'm here to learn. So, hey, I've been here, guys.
Hi, I'm Samantha. I am on my way out, undergrad transitioning to master's. So yeah.
My name is Grace. I'll be starting my master's at TMU this fall, in Mariah Sokolowski's lab. And I'm interested in using neuroimaging methods, specifically FNIRS and EEG, to explore the role of mental imagery in math performance, math anxiety, and like later life outcomes like occupation. Our other two group members aren't here, but I'll give a little blurb on them. So Hillary is going into her second year of her master's at TMU, also in Mariah's lab. She is also interested in neuroimaging and math achievement across the lifespan. And her master's thesis looks at EQAO data and compares grade 3, 6, and 9 from 2013 to 2024, I believe. And then Daniel also goes to TMU. He's a second year undergrad. And he's interested in the applications of machine learning for brain tumor detection. Give you some background on our project. Our project is based on a 2022 paper by Sokolowski and Levine, exploring a specific network neuroscience model called the core periphery model, and how this relates to different neurodevelopmental disorders.
So network neuroscience models often draw upon like graph theory frameworks where system elements like nodes that support like local processing are connected by something called edges. So nodes and edges, you can see, form network communities linked by like these highly connected nodes in the brain network that occupy like these central positions all over the organization of the network. So like you can think of like nodes as like systems or brain regions or tracts, and the edges are like the connectivity. And so in the core periphery model, the core is like this dense subgroup of tightly packed nodes, while the periphery consists of like these sparse nodes, which are like preferentially connected to the core and loosely connected among them. So neurodevelopmental disorders share reduced structural and functional connectivity between the core and the periphery of the brain networks. And so in this paper specifically, the neurodevelopmental disorders that they looked at included deficits across like different domains. In this case, perceptual, mnemonic, and academic. So for perceptual, they looked at deficits in like facial processing, like congenital prosopagnosia, pitch processing, mnemonic ones like deficits in autobiographical memory, like SDAM, and spatial processing. And then as well as academic. So they're looking at deficits in reading, like developmental dyslexia and mathematics, so developmental dyscalculia. And they propose that neurodevelopmental disorders share the common neural substrate of altered connectivity between the network core and the periphery. So these altered patterns may be connections, like the bold lines. They may be enhanced, or they may be like weaker. So you can see how these are like dashed lines and compared to the typical development. And they were thinking that the enhanced connectivity may emerge because of the network reorganizing or compensating. And then we have a 2024 study from Howe and colleagues, and they further explored this idea of varying levels of core periphery connectivity in people with something called ticker tape synesthesia. Do we know what synesthesia is? Have we heard of it? Yes. For those who don't know, synesthesia is kind of like this neurological condition where one sensory input may trigger like a response of like a different sensory, unrelated sense. So like the brain kind of routes the sensory information through another unrelated one. So example, like some people might be listening to a song or like hear a sound, and then it'll like taste grass or something. Different examples of that. So in ticker tape synesthesia specifically, their experience is like when themselves are talking or they're hearing people talk, they kind of see words as like subtitles. So in this study, they collected resting state fMRI data from 17 people who had this type of synesthesia and then 17 control. And they found that these people had increased core periphery connectivity between 5 nodes compared to the controls. And so this leads us to our project.
And so our main research question is, how does core periphery connectivity differ in adults with attention deficit hyperactivity disorder, ADHD? And we do have a definition here for those who are unfamiliar, but it's defined as a neurodevelopmental condition categorized by persistent patterns of inactivity, hyperactivity, and impulsivity that interfere with daily functioning of development. And so the whole rationale behind this is that future research is needed to implement recently developed model-based approaches to uncover whether connectivity between core and particular periphery models of brain networks account for differences between diagnostic groups of individuals. And so to answer our research question, we will be utilizing the Nathan Klein Institute Rockland dataset, which is an open source dataset with more than 1,000 community sampling. It includes a wide array of physiological and psychological assessments. So predominantly, in our case, we're looking at ADHD. So we do have
The sample does include people with ADHD. And it certainly fits within our research question and our research exploration here. The fMRI data is already pre-processed and it's open source, right? It's available. It's the data set that I've actually worked on a few times. Right.
Yeah, so our idea was kind of find a way to implement this core periphery model into the fMRI data set that we have. So for that, we are the first step will be to extract connectivity features using graph theory analysis. So this means that so we have the connectivity matrices and we're trying to summarize all this information, which is like very large data sets, into features such as like the strength of each connectivity or the modularity of each connectivity, and then take these features and evaluate them, evaluate which are the top contributors to differentiate between the groups. So we will have like a ADHD group and a control group using machine learning. So we're thinking for now, probably using random forest to input all the features and then having the classification and then yeah, identifying which ones are the ones that could be used as yeah, to summarize the differences between the groups. So for these, we're using codes available from the Brain Connectivity Toolbox, which allows help us to identify these features to summarize the fMRI connectivity data. And we'll use RStudio, Python, and GitHub for our different steps in the process. So the objectives or the project will be first to use R for data wrangling, so to kind of subset the data and make the classification of the individuals, but also identifying like a threshold for the connectivity. So kind of like to keep yeah, to keep the computational power a little bit low. Then we'll implement the connectivity feature extraction from the toolbox using Python, and then finally explore the machine learning applications for the feature selection. So we will have at the end like our script that we use for data wrangling, the Python script that we use for the connectivity features, which can later be used like in a bigger data set or without subsetting the data. We'll have also a draft script for the machine learning algorithm, and will be all collected in a GitHub repo. Yeah, so for our plan is for the next week, pull all the relevant information that we have about the data set and start complete the data wrangling in R. Then also parallel to that, we'll be working on the extraction of the features, running the core periphery model analysis. And then for the next week, for the final week, just finishing up any remaining feature extraction that we have left. And this also will be mainly the machine learning development. And these are the modules that we're thinking of completing. So first, the machine learning for neuroimaging, the brain data structure ecosystem, maybe the open data and project management modules if we have time, and the data visualization.
Yeah. And so like in terms of like dividing work, obviously 2 of our group members aren't here, so things may change, but so far Lorena will be writing the Python script for extracting the connectivity features. Sam will help with data wrangling. We'll also be doing some data wrangling in R. Hillary will be establishing the GitHub repo and like make sure everything is up there. And then Daniel will be implementing the machine learning.
And there will be overlap in between the tasks
Yeah.
that we're doing, for sure.
That's it. Thank you.
Background, right?
Everyone stop. This one.
Yeah, I feel like this is particularly interesting to me as I don't know if like the author talked about why they think this might is because for this sorry,
Synesthesia, yeah.
synesthesia people, it seems that like there's intense connectivity within the social level prefrontal cortex, if this is from that area. So I wonder, like, why is that the case? Because, like, the DLPFC is not the first thing we think about when we think about like the sensory information connection, the sensory gaining and stuff.
Yeah, and why it's not going through like the primary sensory cortex. I don't know, maybe it has to do with like integrating like information or processing it. I'm not sure.
I see, I was just wondering if
Yeah,
like the
good
authors
question.
talked about it in the paper.
I don't think they did. It was
No,
a
I
short
think-
paper.
Yeah, it was mostly about the strength of the connectivity and how it differed between the synesthesia,
Synesthesia.
yeah, synesthesia and the control. So there was like a higher connectivity happening. But then they divided by like hemispheres and these things, but they didn't go into the
I
this
see, so like,
discussion.
in this case, the area 9 and 46 would be the periphery and the area 30 would be the core. My interpreting is right.
I think so, yeah.
Thank you.
Oh, you're referring to the Rockland data? Oh, it's from my experience, it's a highly reputable data set. I would say yes. I haven't had any issues with it so far. I've done extensive research on past studies that I would have used the Rockland data set. I think a few of us here actually like use it, maybe a few of the TAs. No, just probably one of my recent, like my Charlie would have used. So, one of, my, the little thesis colleague of mine would have done a similar study with that data set, using ADHD. So I think that was the first time she actually used the
Yeah, I
data
think
set, ADHD, but other than that, no. not to
Yeah,
my
but that's a
knowledge.
good point. Like we could explore like what like the publications that have come up from this data set and just like
Yeah,
compare the results to what we get in this
Definitely.
implementation.
yeah.
So if you guys are especially interested in ADHD, how are you accounting for participant motion?
Participant motion?
So to give, so they did it, you're smiling. So a little context, this is kind of a funny story, but when they published NKI, some of the first published ADHD data set created a challenge to say, can you find ADHD from your imaging data? And it found that they could classify ADHD pretty well, despite how much kids moved in the scanner.
Okay.
So how are you accounting for motion?
Good question. I'm not sure, because when we have the NKI, it just comes, I think it's self-reported. It's in the demographics.
Yeah,
I'm not
like
100%
behavioral
sure.
assessment
Mhm.
A bit more in like in the brain.
in the brain.
How are you controlling?
Yeah,
I'm not sure. We're
We don't
open
have,
to-
yeah, we don't have a,
So in pre-processing, yeah, you should covariate, part like the covariance.
Yeah.
Motion.
Yeah. Yeah, so you could be looking like at like the motion, the actual parameters that went into motion correction.
Okay.
And over there.
And then compare it to the ones that have done the studies to account for motion.
so put it into the,
Yeah.
Machine
Yeah,
learning,
so
yeah.
going through pre-process data and find the actual motion records from the pre-processing, like the pre-processing of.
Mhm. Okay.
okay,
You don't need machine learning. It's just in the pre-processing steps
Mhm.
when you're like pre-processing the imaging. This is before getting the connectivity matrix.
yeah,
So
okay.
we might look into that data.
Yeah.
Like right now, we just have like the process data already, so
Yeah.
we can look into the pre-processing.
Did you process this?
Is this the Rockland data?
Yeah, this is the UK biobank.
Oh, Devon. Bake someone from Baker's process with the virtual brain pipeline.
Yeah.
But I don't think they account for motion specifically. Like, at least not different from the healthy control.
Yeah, but every pre-processing algorithm should have a multi correction step.
Yeah,
Most of them now.
I think it's in,
Like, yeah,
I think it's in
first,
full.
Like in.
Just get virtual brain up there, compound file or?
I hope so.
There should be, but I wonder if they need extra steps for motion correction.
But, if a pre-processing pipeline doesn't have multi correction, I'm very disappointed.
There should
My
be.
2002, like, my 2002 FOI pre-processing when I was a grad student, like, oh god, in 2002. Still had motion correction. It had motion correction and smoothing and almost nothing else.
Well, I have a question. So do people, not just children who struggle with attention, who struggle with concentration, always, are they always like hyperactive? Or do you see some that are purely hyperactive, and they're purely attentional issues, and then some both?
I don't know how much. So, yeah, well, like ADHD usually self-report scales usually have separate. Or even parent report usually have separate reports of inattention and hyperactivity.
I think we do have those in the
Yeah.
in the demographic. I think they do distinguish between hyperactivity and attention problems.
Yeah.
Yeah, so you can see it, the hyperactivity. Covered over like social emotion and attention.
But there are studies that show, that, if you scan a kid, multiple times, that, if they move, the first time, they're likely to move again for the other kids. And, it's also heritable because their siblings generally have the same amount of motion. But usually motion decreases as you get older. But it does not, it's not completely gone. And you look at adults with ADHD.
Let's have a suggestion because you are working on a drop data. So instead of manually expanding kids, we can just use some models like RG, the rates that have these problem networks. And also they have these optional expandability modules to understand which like nodes or the other like things on there. So this is one that is really cool.
Okay, yeah, thank you for that. Yeah, good.
Thank you.
Have your cake.
It's payment.
Yeah.
Incentive.
Well, I'll get your names next. Next group is Delay.
Oh, you wanted my name?
Yeah.
Tommy.
Tom.
I'm not actually in IMS, but
Okay. Nilay and Manit. Oh, this one first. I see. What is sorry. Are you muted? Can you unmute? Yeah. I'm going to mute myself and then you unmute. And then, okay.
oh,
Whenever you're ready.
Hello, good afternoon. I'm Ingrid. I'm a first year PhD student in IMS.
I'm Amanda. I'm also a first year PhD at IMS.
And today we'll present our project pitch age-dependent EEG patterns for predicting treatment response in ADHD. So ADHD is one of the most common neurodevelopmental and psychiatric disorders. ADHD is characterized by significant neurophysiological differences that can be observed in EEG recordings. So electroencephalography, EEG, is a neuroimaging technique used to extract features and patterns such as power spectral bands, where there are 5 bands that represent different functional characteristics. For example, the theta band is for deep sleep and beta band for awake and concentration. And the theta to beta ratio is a well-known pattern in ADHD, as well as the alpha peak frequency, which is associated with focus. A promising development in EEG research is the use of AI and machine learning as an advanced signal processing tool. And there's also age-related changes in quantitative EEG in ADHD. However, there is a lack of personalized treatment recommendations based on EEG patterns for EEG and age for individuals with ADHD. So for our project, we ask, how can we use EEG patterns to predict treatment responses for individuals with ADHD across different age groups?
In terms of objectives, the first step, we want to map age-dependent EEG patterns across lifespan by tracking developmental trajectories of key ADHD biomarkers, for example, theta beta ratio and alpha peak frequency. And then we will be trying to associate the identified EEG patterns with treatment response profiles for responders and non-responders. And then for the third objective, we want to predict treatment response based on different age groups and their EEG patterns. The data set we have identified to use is called TD Brain database. It's a very extensive clinical EEG data set with 1,274 individuals that are collected over 2 decades. It has a very wide age range from 5 to 89 years old. It has EEG recordings, neo-FFI personality data, demographic data, and different behavioral measures. We chose it because it's publicly available from Brain Clinics Foundation, and we can just directly download it without any application steps. And it also comes with a Python-based pipeline for pre-processing and generating some of the analyses graphs that are included in the original paper. We thought it's very useful. And its ADHD cohort contains 271 individuals, and there are 102 individuals from their ADHD cohort that comes with neurofeedback treatment outcome data. This is how their data is organized. It's organized in the bits format, so we can just directly use the Python M&E package to process and run some of the analyses.
The tools we'll be using, as mentioned, the TD Brain custom preprocessing pipeline and Python-based EEG analysis, mostly M&E through Python and machine learning. So in our first step, we'll do the preprocessing for filtering, artifact rejection, and segmentation into epochs. We'll stratify the samples into age groups for categories less than 18 years old, adults ranging 18 to 59 years old, and older adults for age 60 and above. Then we'll extract key EEG parameters like the theta beta ratio and alpha peak frequency, as mentioned before. And for our first objective, when we map age-dependent EEG patterns to generalize additive models to capture nonlinear age effects. And then we'll also perform group comparisons using mixed effect models, comparing the treatment responders versus non-responders. For our second objective, we'll correlate the EEG patterns with treatment response, and we'll do this with multiple regression and PLSC analysis to identify patterns from EEG that co-vary with treatment response. And then for our third objective, where we predict treatment response, we'll use machine learning, specifically random forest model, using EEG parameters and age to predict neurofeedback response, and we'll use a bootstrap validation. Our deliverables will have, by the end of this project, a GitHub repo, our project reports, and time permits interactive, our Shiny dashboard.
And then for timeline for week 3, we're both me and Ingrid are going to be performing data pre-processing and to extract the EEG parameters that we mentioned before. And we are also going to visualize the EEG patterns across different age groups, comparing treatment responders and non-responders. And for week 4, I'll be conducting regression association PLSC analyses to correlate EEG biomarkers identified to age stratified ADHD treatment response profiles. And Ingrid will be building machine learning based treatment response prediction models. And the related BrainHack modules are introduction to R, introduction to deep learning, the Python M&E package with EEG BIDS data, and introduction to partial least square and machine learning for near imaging. Yeah, that's everything. Thank you.
Based on like the power bands that we'll be extracting like these parameters, we're expecting to see some changes between the different groups. That's kind of how we'll be able to evaluate our outcomes based on extracted from the raw data of EG. Yeah.
Or like, are you guys using any other line to extract these features? Because you have like 200. With the sample size, because we're specifically evaluating treatment response, only around 100 samples have those data. So we'll be only working with that data set. And I believe for the Python package called M&E BIDS, it is able to extract those biomarkers we are interested in. So like everything is automatic. You don't do like manual rejections of artifacts. You mean? Yeah. Those artifacts removal comes with like we mentioned before, this data set comes with a pre-processing pipeline in Python, and that pipeline is specifically built for artifacts identification and removal. So that's like during the pre-processing step.
We'll most likely just have to set the different thresholds to our purposes for this project. Yes.
It's like, right. With all the ROIs, like are you guys using the grand average or like?
Well, I mean, hopefully this answers your question, but like there's 19 electrodes that we're working with from this EEG that was part of this data set. So we'll just use as much as we can in this project. So the whole thing, yeah.
That makes sense. Thank you.
Yes. Okay.
I'm Kayla. Okay. So just curious, treatment response, is that defined by a certain kind of outcome in the data that they already provided? Is it, especially across such a large age range, like what does response look like for someone who's 5 versus someone who's like?
In the sample data sheet, they are specifically evaluating the treatment response for neurofeedback therapy. They have a measurement for 2 different subscales, attention and hyperactivity. They have treatment measurement. It's like a score, and then post-treatment measurement, and the the difference between those 2 scores would be used to define like responders versus non-responders. Yes.
So I don't know if you mentioned this, but like, since you're looking at age-related changes, are you like categorizing the ages, or are you looking at it on like a continuum?
Yes, we're gonna categorize into 3 ages: below age of 18, 18 to 59, and above 60. So.
Any specific reason?
Yes. Just the way, because we have like there's over 200 samples, so then we did like categorize by like the decades, and then just ended up being like 50. Tried to keep around like 50 to each category, so I ended up being 18 to 59, and above 60.
So why you would have just used the continuous age variable?
We Wait, I think in the method part, for the developmental trajectory, we are planning to model the age as a continuous variable, and then we wanted to do like group comparisons and then break them down into 3 large groups. Yes, no.
Make sure that the changes in power are actually not just changes in the overall background noise, which should be spectrum axis, but it's actually changes. So so once you have your spectrum power spectrum produced with the MD pipeline, you can just go to group and it'll give you those as individual values that will be considered well to using the spectrum. Peak frequencies for each opera wave.
I also want to mention that I'm sort of in this space and so beta ratio has been sort of disapproving. So I'm just gonna mention more if you would consider other features to include in them, since it's not like alpha is considered a character of static system.
I think the parameters that will come out of this toolbox as well.
Okay.
No, just on the end comment that like after applying code, you really like you never have this like you really only have like beta and alpha. But like I think alpha frequency people are really interested in. I think the strongest. Beta will depend on is this resting state?
Yes.
Yes.
So is it nice open and nice things and stuff?
Yes.
2 minutes of each.
But then, so the freedom response, that's from about 100 people,
Yes.
and you have all 3 age groups represented in the treatment response data.
Yes.
Okay. Yeah, I also had a comment on that, just like whether you will have enough power to see because between the groups, if you have 3 groups in 100. Like example.
Yeah, we are, I think we are also thinking about that problem because either like cross-validation or like bootstrapping needs a large sample size, especially for building machine learning based models. So we think of it as more of a exploratory analyses, trying to see which features are predictable or not to like use it for a recommendation or trying to classify patients. That works. That's better. That's fine. That's better. Okay. Got it. All right. Are we? Yeah, I can start. Okay. Hello, everybody. So our project is going to be focusing on increasing the accessibility of open access brain age calculators. So just some background, normative brain age modeling is a powerful tool that allows us to look at how an individual's brain deviates from normative reference groups that are age and sex matched. And they output two different types of metrics. The first is a centile score, which, like I mentioned, is a deviation. And then there's the brain age gap or brain age, which you'll hear a lot, which is the difference between a chronological age and model predicted age. There are a number of different open access calculators out there, and it's a great way to kind of quantify and different brain health metrics. And yeah. So there, like I mentioned, there's a number of different calculators, and there's a number of different characteristics that they differ on. So the first one being training data. They can be trained on a variety of different types of data sets that have and they can be different sizes and demographics, specifically age. And they use different machine learning models, which is why there's different brain ages that are calculated, even though they kind of have a similar method. They also have different input features. They require different input features. So this can include cortical thickness, surface area, volumes, or subcortical volumes. And these input features can use different free surfer atlases. And so these and are used to obtain different input features. And lastly, like I mentioned, they can output centile scores or brain ages. And so because there's so much, actually, yeah, that's the third part. Yeah, sorry. Yeah, so as Gia mentioned, there are a bunch of different calculators out there, which are very useful, but there is limited accessibility because there are a few problems that people run into. So for example, each calculator requires different input information, as well as it requires different like MRI metrics. So it can make it kind of difficult to identify which parameters need to be input, as well as when there is an issue with the inputting the data. There's no error message that arises, making it kind of difficult to troubleshoot. And so for that reason, we want to create the centralized Git repository with data wrangling scripts that can populate the calculator specific templates using outputs from the T1 structural MRI images. And so the end goal would be to hopefully streamline the process of using these calculators as well as promoting their use. In terms of tools and methods, so we will be using the reproducible brain charts data, which is an open access platform that has imaging data from a number of different data sets. And specifically, we hope to use the developmental Chinese Color Nest project, Healthy Brain Network, and Rockland sample, all of which are openly available in reproducible brain charts. And the reason we're using reproducible brain charts is because they have all of the pre-processed data. And like I mentioned, there's a number of different input features that are needed for different calculators. And so this way we don't have to do the pre-processing part of it. And they also use the different atlases as well. And in terms of the calculators we will be evaluating, we are focusing on these 7 that I have listed. This kind of shows the training data and input brain features that are required for these 7. We are going to look at whole brain calculators as well as ones that output tissue specific, region specific, and network specific scores. Yeah, and we will be using R to write the data wrangling scripts.
So we have 2 deliverables with this project that we hope to achieve by the end of the course. The first one is writing the final report and as well as composing the final presentation. And this will be a collaborative effort that we will all do together, which will be detailed by Eleanor in the next few slides. And the second one is that we hope to create a public GitHub repository where everyone can access the code. So hence, if anyone wants to work on the code further or utilize it in a different aspect, it could be easy to do so. And so basically the timeline of our project, so what we've achieved in the first 2 weeks was, in the first week, we've completed relevant modules to help us be successful in this project. And we've also come together to brainstorm more about this project as well. We've also defined project goals and outcomes in the second week. And in the weeks that are coming, the third and the fourth week, we hope to download the RBC data and modify, first write the code and then modify it. And then in the final week, we hope to troubleshoot the data, ensure that everything is running smoothly, as well as compose that final report and do that final presentation.
So we divided the work among team members, and we plan that each person will work on like data wrangling script for around like 2 calculators, and then we'll work on different parts for the final presentation. And I feel like the report we will write it by ourselves. And then these are the related BrainHack school modules that's related to this project, including Git and GitHub, R, machine learning basics, and for neuroimaging.
Yep.
So in terms of like how you would apply this in like a clinical setting, what kind of population would you be able to like look at?
That's a great question. So I think BrainHack calculators are being used in a number of different ways, even just like amongst the 4 of us, we have different ways of using it. It's a good way to compare just a clinical sample versus what the normative reference group is like. So it's interesting to look at, for example, in my research, we look at individual psychosis spectrum symptoms. We look at whether we see deviations in their brain age versus age and sex match controls. And I think in a developmental sample, it's interesting to look at to see how their brains just compare to someone else their age. And it kind of applies. But yeah, if you guys want to go and explain how you're using it.
Yeah, like similar to Gia, I work with like individuals with schizophrenia spectrum disorders. And so I'm specifically looking at brain age in these populations and how it relates with visceral adipose tissue. So that's kind of how we've been using it. I don't know.
So I'm using it for psychotic depression. So just to kind of add on what Gia said, brain age is really versatile. So it kind of creates a trajectory of someone's entire life, and you can use their score or the brain age that they get against a normative population. So basically, if someone is like 25 years old, but their brain age is 30, you can use that against a population of people that are already 25 years old. So it kind of gives you like a benchmark, which you can use to then be like, okay, this person's brain age is a little bit higher than normal. There may be some underlying issue there.
So I guess as like a follow-up to what you were just saying, I guess let's say that someone's brain age is older than it should be. I guess how would that inform the type of, I guess, treatment or intervention we would provide for them?
Yeah, so the treatment would be something that would have to be explored further, but it could definitely, like for instance, for schizophrenia, so like what Isabella is looking at, and for me, psychotic depression. If we're looking at a population where we see that the brain age is quite advanced, that could inform of some potential diseases. So it could inform us that there is some investigation required into this patient further. So then they would obviously require more like specialized interventions to make that confirmed diagnosis. But really, brain age is a way that you can really ascertain within the population who's having a more rapid brain development, and by that extension, like who requires a more specialized intervention. So it can really help you catch brain developmental disorders before they become so severe that they can't really be treated. Yeah.
So the model that you guys are building, how would you anticipate, or what do you expect the impact it would have in implementation? If it would make it easier for like standardize across different clinical settings, or also just for people day to day? Is it something that you want to make it like publicly accessible to everyone? How would you want that to be?
So for myself, like for my study, it is something that we try to like, hopefully the ultimate objective, obviously it's like one kind of leg in a very long race, but it's something that we would obviously want to implement clinically. Because, for instance, researchers that investigate brain age or like stem cell brain scores, when they look at this, their ultimate objective is to have it standardized the same way as like when you would go to a pediatrician's office, you would see like the trajectory of a baby's weight or their height and how that like what a normal baby's weight or a normal baby's height should be. And if your child has abnormal weight or abnormal height, or if they're not developing that well, that can inform you as an individual, but also the clinician as well. So that's kind of where the direction of both BrainAge and Central Brain is hoping to achieve within the future. They just kind of want to standardize so that it's kind of a metric that everyone can use for themselves the same way that we do for like height and weight at a pediatrician's clinic.
I just wanted to add on though, we're not building our own model. We're just going to be using ones that are already available, and we're also not standardizing any of it. It's more so that we're trying to make it more easy to use them. But I do think that like future directions, maybe not for us, but you know, for the individuals making these calculators, is to create like a standard calculator that's, you know, maybe the most optimal performing, so that we could, like Kubate mentioned, do all of those things. But we're more so just trying to see, we're just trying to make it easier to use them so that they could be more widely used just by a regular researcher that might not be able to, you know, because they can be really complicated. And if you have a really large sample size, it can be difficult to manipulate the calculators. And so we're trying to make that easier. And then hopefully if we can make that part easier, more people use the calculators and see which ones are performing better and which ones are the best ones to use. So we're kind of doing that part of it.
And even like very like immediately, I know us three are going to be like get benefit out of this because we're all using those calculators for our thesis. So that would be nice.
Doesn't.
I think it just did speed, right?
Oh, great.
We could try mods up, but
Yeah.
where did it crash?
You just treat.
Good.
It's no.
No.
Oh, wait a second. I need to.
Oh,
Oh.
Okay,
Yeah,
oh,
it works.
going on pause.
Thank you.
You know where to kind of work? I know.
Go ahead. Whenever you're ready.
Okay, so hello everyone, I'm Lucas, and our group will try to investigate a deep learning based localization of EEG electrodes, and we will start from a 3D point cloud object.
And I'm Joel Yev. I'm a third year PhD student in the IMS program, but not doing it for credits.
Okay.
Which is why I can't make it to the my list. And we have another member, Ivy, but she's out sick today. So background information, is anyone familiar with like EEG processing in general? Well, with EEG caps, there's a sensor on basically like a swimmer's cap. Someone showed the picture of the EEG cap earlier. But most caps come with like a template model, but everyone's heads and head shape is different, and the placement is never really exactly the same. So whenever people can, we collect custom electrode location maps. So essentially location maps that are customized for each participant, and that takes into account differences when you do comparisons across groups of people. It's essential for accurate source localization and modeling. If anyone's familiar with source localization, which is essentially taking the brain signals from the channel level and projecting it onto the brain and trying to determine where the source of the signal is coming from. So conventional EEG digitizers are really expensive. If anyone's worked with like the Behemoth one, that's around $10,000, and you have to manually pinpoint each one of the electrodes, which takes some time with the participant that you can spending actually recording data. So most people are using newer technology nowadays, sort of like video grammetry or handheld 3D scanners to essentially scan someone wearing the EEG cap. It only takes a minute, and then doing all the digitization afterwards. The issue though is that you still have to manually label each one of the electrodes to get the 3D space, the head map. So our research question, or our question is it possible to develop a protocol with deep learning to automatically label EEG electrode coordinates that is generalizable to different EEG setups and data types? The data set that we have is from this EEG simultaneous EEG fMRI study that I've worked with in the past. It's available in the National Data Archive, but will be available on OpenNeuro eventually. But it's alpha oscillations of working memory deficits in ADHD. It's a multi-imaging study. I'm going to mention it now because the next group will be actually using this data set. So I'm going to describe it a little bit. Essentially, what this project's all about is looking at alpha markers that we found in ADHD, specifically alpha event-related decreases during working memory processing, and how it relates to functional connectivity. So we can determine what is actually going on in the brain that relates to these EEG signatures that have been shown to be reliably different in ADHD controls. And this data set includes EEG recordings, simultaneous EEG fMRI recordings, and DTI as well. So it's a pretty rich data set, which will be eventually available on OpenNeuro, just not right now. But what we're really interested in is the 3D scanned head models that were used to get custom head maps, as well as a portion of the data that actually scanned T2 weighted MRI images. So if you see here, this is actually a picture of me with the EEG cap on, and you can see in the T2-weighted image, each one of these sensors are lit up. And then this is like a 3D rendered version of it. And so the idea is rather than manually selecting each one of these sensors, like this took me 30 minutes to do, can we automate it to figure out where each one of the electrodes are and then actually label them? And so the tools that we're interested in that we're hoping to use is some Python machine learning tools, specifically this Open 3D library that will convert the MRI and 3D data that I mentioned before into 3D point cloud format. And then again, this is because we want it to be generalizable across different data types in the future. And so, and then we would be using PyTorch and PointNet to sort of fine tune the learning model and to test it, the accuracy later on.
So the objectives, as Joel said, we want to automate the electrode localization and labeling, and we'll try to do that with the deep learning model. And also we want to make sure that the pipeline that we are going to build is accurate, has accuracy between the same data, which is the OBJ, and also different with the T1 weighted MRI images as well.
It's T2 weighted.
Oh, T2, yeah. And also to be able to create a pipeline that is accessible to others to use. So when they are using other kinds of data sets or setups, so they can be able to replicate the pipeline as well and train in their own data sets. So what we want to deliver is the GitHub repository where we have the scripts and also the documentation for training the model on different data sets so others can use. And the timeline, we plan next week to prepare the data set. So we need to label the electrodes first to be able to fine tune the model and also fine tune the deep learning model and data sets. And then on the week 4, we will check for the accuracy and prepare the documentation and guide so that we can create the repository. And the modules that we are planning to use for the project is the Git and GitHub and introduction to Python for data analysis, and then also the introduction to deep learning and Python packaging. Yeah, thank you.
Yeah, very cool, very messy though.
Like where does like the ports connect? Like so you'll have to like where like and there will be like ports connecting to the cap and to like the
it's a EEG amplifier, but it's encased in a MR compatible casing to essentially make sure that doesn't get pulled into the MRI, but also to like reduce the amount of noise that goes in. But yeah, we collect EEG outside first and then inside because we need a ground truth of like, does our data cleaning mess up our signals, our ultra signals? So we record outside and inside the center. But yeah, it's really messy. But helpful, sort of actually. No, we're moving away from doing it, but it's helpful if you want to understand an EEG signal and what brain region communication is occurring to generate that signal. But nowadays, yeah, the F near is EEG, so I would argue a lot cleaner.
In terms of cost and like just the setup time, wouldn't it be more efficient just to use MEG rather than simultaneous EEG and fMRI?
Yes, but not every institution has. And it's really expensive upfront to purchase a device. But now I've heard they have like portable MEG.
Wow.
But it's still being like tested. I think it's like OMP. OPM.
Wait, does it get
Yeah. But they're not that good. They're not as good as actual. And they still require half of what? How big is the room that cancels out the Earth's gravitational field? Yeah, and it's under in the parking garage, which is.
So basically, if you can't afford a Meg, you sort of use, well, yeah, I'm trying to see how much it actually costs, because you think like Meg is still more expensive than simultaneous MRI and EEG.
Yes.
Yeah.
But it also has worse like temporal resolution than
MEG? Yeah, no, it has better temporal and spatial, but not as good as region simultaneous MRI or MRI.
fMRI.
So simultaneous fMRI fMRI has the most spatial resolution.
Yeah, Meg is really just like giant bucket, and then you actually put it's giant bucket, and you put the EEG cap on the head while the giant bucket is scanning yours. So I think it should have ceteris paribus if you use the same number of electrodes below the giant bucket and versus the EEG. I think you should have the same temporal resolution. Well, yes, but how come it's better than in EEG?
Temporal resolution.
Yeah, like why is it better? Like the it's not degraded by when you travel through the dura matter, and then this. Oh, right, that's yeah, I think that. So better signal.
And when this was all collected in Los Angeles at UCLA, and whenever we inquire about MEG, they'd be like, "Why don't you send your participants to San Diego? We have an MEG device."
Well, they have one that's like kids, no?
Yeah.
Yeah, I mean, yeah. In theory, we would use it, but I'm not sticking around long enough to try to set that up. It's a mission to.
Oh yeah, you have like EGF.
Oh, sorry, I just mentioned that because
But potentially as like a cheaper
alternative.
Alternative.
Yeah, mostly if you already know your EEG signal of interest, because you're limited on the number of channels. Yeah, any questions about the project?
Have you looked more into the types of models you're going to use, or are you starting from a pre-built model and you're fine tuning it, or?
So we're good. We're actually going to be using it's called the point net. It's like PyTorch. Actually, you looked at
Yeah, so we use PyTorch to access the point net, and then we can fine tune. Point net has many neural networks that we can use, and then we use the PyTorch to access those, and then we can fine tune deep learning model from there, and yeah.
So it becomes a image segmentation problem, because now that you convert your 3D model to individual points, you would just label each points, like we would label these bumps as electrodes and then everything else as scalp, and then train the deep learning model to find these bumps on the head, and hopefully learns a pattern from the 128 channel caps that they should be somewhere near here. So that it could be robust, and hopefully also work on our a separate type of data that has the same cap, but it's I assume that the 3D point cloud is going to be a little different.
Great, but like, what is the best contrast for like EEG cap versus scalp? Or is there like, do you get good EEG cap versus scalp contrast?
So this is the EGI 128 channel net. If anyone's familiar with that one, it's essentially a bunch of sponges, and you like dunk it in water, put it on someone's head. And because of the way it's set up, the sensor is actually raised by a few, like 2 millimeters, I want to say. So it gives a nice contrast between scalp and.
Yeah.
So it'll be interesting, because now I'm working with the biosimilar device, and it's a lot closer to the head. So I'm hoping after this, if we, if it works, I'm trying to apply it to the data I'm collecting now. And see if it's still as accurate. Another thing is, what we could also do for that is also include RBG, sort of like color into the 3-point cloud. I don't know how exactly yet, but and hopefully that'll help the model, because for that system, there's a contrast between sensor and cap. Hopefully that would improve the model. Present.
Should we just present the review? You don't think we have?
I think we should another one. And go back. Is this good or?
To full screen, like click on the dot dot dot, and then you could go into full screen. Where is it? Mark.
Oh. Doesn't show here.
I did that. And then go slideshow. So, hi everyone. Our project name is Functional Connectivity in ADHD, Group Differences in Predictive Modeling during Spatial Working Memory Task. So I'm Nilay. I'm working as a lab manager in Toronto Metropolitan University in Caspion's Memory Lab. And I will start my PhD in University of Alberta next year. I will focus on fMRI data in autobiographical memory. So I'm here to gain some experience. I'm a beginner, actually.
And hi everyone, my name is Manit. I'm going to my 4th year at York. I'm in my undergrad and I'm majoring in biomedical science. And I'm currently looking for research opportunities.
So the other groups actually gave some basic information about ADHD, but here we will focus on more about spatial working memory in ADHD. So patients with ADHD differ in frontoparietal circuitry associated with working memory, and spatial orienting and effects of load working memory have demonstrated lack of some differences, and these findings show further study of brain mechanism that contributes spatial working memory deficits in ADHD. So therefore, we want to compare functional connectivity between ADHD and the control group and predict the diagnosis of ADHD using some machine learning with the spatial working memory task-based fMRI data.
And in our project, we're tackling 2 different questions. The first one is, how does the frontoparietal functional connectivity during spatial working memory tasks differ between adolescents with ADHD and with neurotypical controls? And the second one is, can ADHD be predicted using machine learning, which has been trained on functional connectivity data sets during the same work?
So our data is same with the last group, actually. We were in the same group, but then we separated because they want to focus on more EEG, but we want to focus on fMRI. So this is a multimodal data, as they said. So it includes 140 young adults, and it is a pre-processed data. And yeah, it includes a computerized version of the spatial working memory task.
And the tools and methods that we're going to be using for this, first is Git and GitHub, which will be used for visual control. That's where we put our repository. Then we'll use Python libraries, which will make things easier with Scikit learning for machine learning and learning for fMRI connectivity analysis. Then we'll be using PyPod, Body Express, and C1 for data visualization, and lastly Jupyter Notebook, which will probably be posted on the Indeed.
So our objectives here, I mentioned here our personal objectives. Actually, the main goals of this project is in general learn how to work with fMRI data, and more specifically, we want to get more practice with machine learning tools for fMRI data, and we want to learn to compute correlation matrices and perform some group comparisons. As I said, we both are beginners, and we want to learn these things.
And what you can expect from us at the end is a GitHub repository with all the code scripts and data preparation. We'll also have figures and demonstrations and results, which will be summarizing our performances of the models. And lastly, a Jupyter notebook that includes all the codes and videos. And for the timeline, we have divided it into 3 and 4. First, we'll be catching up on all the modules. We have just wrote data visualization with mainly all of them. We'll also be going through data prep and analysis, which I'll be focusing more on, while Nilay will be focusing more on machine learning part of the project, which is very generalized, but we're going to be helping each other along the way. And then for week 4.
So week 4, our goal is hopefully complete the group comparison in terms of functional connectivity, and we will do some data visualization, and we will prepare the presentation together. And as he said, we will support each other in the process.
And lastly, the BrainHack module that we're going to be using. We did list a lot of them, but the main thing that we're going to be focusing on is machine learning basics, machine learning with neuroimaging, fMRI connectivity, and introduction to data visualization with Python. But all of them are pretty helpful.
And these are references. Thank you for listening. Okay.
But oh, so I'm not sure if they mentioned it before, but it's just so those spatial working memory tasks can be sent during this scan the child.
During the scan, yeah.
And so I guess my question is like, what brain regions would you want to focus on?
Yeah, we want to focus on the frontoparietal connectivity, actually. I don't know how, but like we want to select this part because it's like important for the special memory task.
So I guess the idea is to do like a case control comparison, or do you know what kind of machine learning model you're training?
So these are our questions, and these are the like separate questions. First, you want to compare the frontoparietal functional connectivity, like using the matrices, and the other part of the project is using machine learning tools separately, actually. Hopefully we can do both.
Yeah. Not sure if the demographic information is available with this data set, but if it is, are you guys interested at all in looking at sex differences in the population?
It would be good. Yeah. Since it's like special working memory, there are like significant differences between male and female. It would be good. we can try if we have time. Yeah, thank you.
The data set is part of the longitudinal study, and I'm pretty sure the longitudinal data set there was about a 50-50 ratio and so like that. But I just don't know how many responded and actually decided to come on the study. But good question.
We have motion directors to bring it to the conference.
Yes, so the data, like, I forgot what it's like an ICA based, a little bit like training the ICA model on like the data set
Yeah.
on it, and then there are motion, various motion activations that can include accuracies.
Okay. But I guess the idea is to like buy some to this.
Yeah, that's it.
Sponsors.
I think yeah, there's the motion estimates after
cleaning. Okay. Yeah.
Yeah, that's why I left earlier because I worked with these data sets before.
They're like, oh, they do so.
You have to like, you have to bring in a child and then go into the mock scanner and then be like, all right, practice staying still. And show the parents that so the people practice at home and then bring them back in. And then cross your fingers and get out the daily. I mean, yeah. There's a meme of like people with anxiety and people with ADHD and they're high-fiving and it says like shaking.
Yeah, but it is a pre-processed data, right? And it's ready to. Okay.
Which is, sorry, I'm going on a tangent. It's interesting because then it really brings to question like if you remove a lot of data, or prior studies that removed a lot of data, are you biasing your sample because you've removed a lot of hyperactive versus the rest? So,
I think there's also a tech component in it. Presentation.
it's back in the day there were higher rates, but that's because some presentation is different. They were underdiagnosed. But women with ADHD tend to have higher tolerabilities of depression and other anxiety.
Good.
Okay.
It's fine. They're trying. They are.
Hi, can you guys hear me?
Yes.
Yes. Okay.
Do you want to share your video or?
I can. There's a lot of people walking behind me, so it might be distracting. I'll stay with sound for now.
Okay. Sounds good.
Okay, cool. So for this project, for Dean and I are looking at the EEG-based monitoring of cognitive states in athletes. So, do you want to skip to the next? Yeah, perfect. So primarily at EEG recordings, and we want to look in an athlete population because this population like operates under high pressure conditions. There's lots happening in especially their frontotemporal plane, so in their frontal networks, and we want to analyze what's going on. So mainly EEG frequency bands along with some other performance measures. For this project, we'll mainly be focusing on the frequency bands to look at changes in relaxation or distraction monitoring, active thinking, drowsiness, fatigue, and different high-level processes. Can go to the next slide. There we go. So the main goal is to analyze the EEG frequency band dynamics. In athletes in general, we have a specific population we'll be looking at right now. And we're going to be doing this while they're performing motor tasks as well as mental tasks, so we can detect these cognitive state changes, and this will help to inform their future training, what different protocols we need to change per athlete. And we are working in a professional athlete setting, so a lot of these athletes have high-level coaches working with them, and this is more of an objective stream of data that we're going to provide to them. So we're looking to take that EEG data in this school and analyze it in different ways using multiple different methods.
So our data is EEG recordings, and then we first have to start pre-processing it. So getting rid of any frequency bands lower than 0.5, anything higher than 40, and we have to do some artifact removals. And then we plan on segmenting it based on what type of tasks they've been doing, and then just analyzing the frequency. So this involves extracting each type of gamma band, and then finally just visualizing those and comparing them between each other and seeing what we get. So this is our expected timeline. First week, or week 3, would be us pre-processing the data and segmenting it, and then week 4 would be hopefully analyzing it, comparing it, visualizing it, and then at the end of it all, presenting and writing about it. So our deliverables include the GitHub. We plan on using Python, using packages such as like scikit, NumPy, Seaborn, and a couple others. And then we'll have it all on a Jupyter notebook, so we can have like a clean pipeline and then the visuals in order. And then finally, we'll have some documentation. So the purpose, the steps, results, and an overall like summary / conclusion of what we did and what we got out of it. These are some references. That's it. Oh, yep.
It may have been mentioned at the beginning, I might have missed it. Are you guys focusing on like a specific type of athlete? Like, I guess what I mean is,
Good.
yeah, sorry.
Yeah, no, yeah, good question. I'm assuming like you're wondering what specific settings they're going to be in. I have some data collected already, and this is in golf athletes as well as court sport athletes. But instead of focusing more on the sport, we're actually focusing more on the cognitive domain or the mental state. So looking at like distraction or looking at higher executive performance. So we're going to see professional golfers. I have a couple of golfers that I have data collected on, and then a few more that want to have this data. So I'll add on to that as well. Like there's a bit of a caveat with how much information we can share at the end of. There's limitations to how much of their personal information can be on the web. But we're gonna like at least be able to, how do I say, exemplify what we're doing through one specific example. Even if we can't provide every single person's data, we kind of just want to create like a pipeline and a representation of how we would analyze this type of data. Does that answer your question?
Yeah, more so like I guess what I was curious about is I guess if you were to include, let's say, athletes who play like high contact sports, if it was like very like general, would you be excluding people who may have suffered like a TBI or something like that? Because that could obviously affect their performance on these types of tasks. But since you guys are mostly golfers and court sports, I think it should be a problem.
Yeah, there's definitely a subpopulation like concussion or even different neurodivergent, different like someone who's just not in the same category as your average athlete. So that's something we'll tackle later down the line. Right now, we just kind of have we're grouping athletes with their skill level within their sport. So whatever sport we choose, we're really looking for the same grouping of athletes just to look at like a standardized data set. So we would either look at amateurs or pros, non-concussion or concussion to be able to create kind of a standardization for that type of population.
I think, I mean, I'm not very familiar with EEG, so maybe like I could be wrong about this, but like from the very basic literature I've done, so I've seen like a good percentage of it use 40 as the upper limit. So that's what I've like subjectively chosen. I think like we could definitely do some more research to figure out a better upper band, and then like change it as it is, but like yeah, that's the best response I have right now.
Yeah. Well, like, since gammas are from 30, I don't know if it would be like helpful to like include a little bit more, maybe 30 to 50 or 30 to 45.
Okay, yeah, we'll definitely put that to consideration.
I think I mixed it, but do you have any, like what EEG setup were you using? Was it like a mobile EEG device? Or like a full head?
I believe it was a mobile, like a headband.
Yeah. And this is like during sport then?
Yeah.
And before and after? Yeah.
Yep.
Was it the Muse?
I don't know. Sam did it, but you can ask. She's muted right now. I don't know what that means.
Oh, sorry, I didn't realize I was muted. I was wondering what the question was. I can't quite hear.
Is it the Muse? What is the?
Oh, for the collection device?
Yeah.
We have two. We have one in a lab setting, which is a full head EEG, and then we have a Muse. We've primarily used the Muse. We have another device that's not as good. And those are the ones we would use in a non-lab setting.
So are you pre-processing all, like, do you need different pre-processing pipelines for the different setups?
I would assume so, yes. Yeah.
Oh, okay.
Yeah. Okay, so can everyone hear me? Turn off. Okay, I'm not gonna turn up the volume, so there's no yeah. I'm gonna click on that. I don't. So this is my one of it's the data from one of my labs. So I can't share it. So could you stop the recording? Yeah, it just cannot be shared online. So it can be yeah.